Plant hormones. Plant hormones, also known as phytohormones, are chemical substances that regulate plant growth and development. They play crucial roles in processes such as cell division, elongation, flowering, fruiting, and responses to environmental stimuli. Here is an overview of the main types of plant hormones and their uses. Auxins. They are made in the meristem tissues of shoots and stems, then translocated downwards to the rest of the plant. Auxins are water-soluble, they easily move through plant cells. Auxins promote plant growth by promoting cell enlargement and elongation. The effect of auxins is best observed at the zone of elongation in shoots and roots. High concentration of auxins in roots inhibits growth. They are required in small amounts in roots. Key functions of auxins. Auxins promote cell elongation and division. Parthenocopy. Auxins are used to stimulate the development of ovaries into mature fruits without fertilization. The development of ovary into fruits depends on the production of auxins by the germinating pollen tube. Spraying auxins on flowers causes blooming and fruit development, such as in pineapple, seedless citrus, and hybrid grapes. In fruits, auxins prevent fruit fall. Auxins cause apical dominance, where the terminal bud promotes elongation of the main stem and inhibit development of lateral buds into branches. Auxins cause stem thickening. In stems, they are produced by the cambium. Auxins stimulate cell enlargement and elongation, causing stem thickening or secondary growth in woody stems of trees. Development of adventitious roots from the stems. Auxins are involved in the formation of roots on stem cuttings. Auxins prevent leaf fall by preventing the drying up of the abscisian layer of cells near the base of the leaf petiole. Low concentration of auxins in roots stimulates root growth through enlargement of cells next to the root meristem, causing root growth. High concentration of auxins inhibits growth of plants. Spraying unwanted plants with auxins is used to control weeds. Influence the differentiation of xylem and phloem. Regulate phototropism, growth towards light, and geotropism, growth in response to gravity. Phototropism and gravitropism. Auxins help plants respond to light and gravity, directing growth towards or away from these stimuli. Practical applications of auxins. Production of seedless fruits. Delaying fruit fall in orchards by spraying auxins, thus facilitate economical harvesting. Farmers prune crops to remove apical dominance, thus encourage development of side branches instead of upward growth. This increases yield and also makes it easy to harvest crops. Weed control. Vegetative propagation of horticultural crops. Auxins are used as rooting agents for stem cuttings for hybrid roses, apples, and oranges. Young plants are sprayed with plant growth substances to stimulate rapid growth. Stem thickening in agroforestry. Gibberellins. Key functions of gibberellins in plants. They stimulate growth. They work with auxins to stimulate cell division and cell elongation. They stimulate the growth of stems with little effect on roots and leaves. Causes increase in height of the plant. Promote seed germination and break dormancy. During germination, the embryo releases gibberellins that mobilize stored food for development of the embryo. Influence flowering. Parthenocopy fruit development, which is the development of ovaries into mature fruits without fertilization. They inhibit rooting of stem cuttings. Practical applications. Gibberellins. Increasing crop yield. Gibberellins application can increase the size of fruits like grapes and apples. Breaking seed dormancy. Gibberellins are used to promote seed germination in seeds that require specific conditions to break dormancy. Used to make dwarf plants grow normally. Malting of barley. Gibberellins are used to accelerate the germination process in barley seeds during beer production. Cytokinins. Stimulate formation of callus tissue over wounded or damaged tissues. Work in conjunction with auxins to control tissue differentiation in roots. Promote cell division and shoot formation. Delay leaf senescence or leaf aging. They promote thickening of stems and roots. Nutrient mobilization. Cytokinins help in the translocation of nutrients, enhancing overall plant growth. Practical applications of cytokinins. Tissue culture. Cytokinins are essential in plant tissue culture for promoting shoot proliferation. Delaying leaf aging. Application of cytokinins can extend the shelf life of green vegetables and cut flowers by delaying senescence. Abscisic acid, ABA. They inhibit growth of plants. Induces seed and bud dormancy. Promotes abscission. This causes shedding of flowers and fruits from trees. Promotes leaf senescence or leaf aging. Regulates stomatal closure during water stress. Practical applications of abscisic acid. Enhancing tolerance of crops to drought. ABA treatments can help plants withstand drought conditions by inducing stomatal closure and reducing water loss. Long-term seed storage. ABA is involved in maintaining seed dormancy, preventing premature germination during storage. Ethylene. Regulates fruit ripening. Promotes leaf and flower senescence. It inhibits stem elongation. Involved in the response to mechanical stress. Uses. Fruit ripening. Ethylene gas is used to ripen fruits like bananas, tomatoes, and avocados. Flower industry. Ethylene inhibitors are used to prolong the life of cut flowers by delaying senescence. Agricultural stress management. Ethylene can help plants respond to various stresses, including mechanical damage and pathogen attack. Florigen. Stimulates flowering.